So probably we can start.
Okay.
And so we were glad to invite Alison Leaf, Dave Roberson from Seven Bridges. They are really excellent. We have been working on the Seven Bridges Cloud on the BioData Catalyst projects. Previously, I worked on the Cancer Genomics Cloud, which is also a Seven Bridges cloud computing platform. It's really awesome. So today they will have a workshop and talk about the NHLBI BioData Catalyst. That's a cloud computing platform. I hope you will enjoy. So Alison, Dave, it's your time.
Okay, thank you so much, Zai-Chen, for inviting us to speak. I'm Alison Leaf. I'm a senior program manager at this cloud computing company called Seven Bridges, and I'm going to present with my colleague, Dave Roberson, who is a community associate. The title of our talk is Democratizing Access to Data and Compute on NHLBI BioData Catalyst, a cloud computing platform. The purpose of the 1-hour session is to tell you about how computing on the cloud can enhance your research. In particular, during this session, you're going to learn about a new cloud computing ecosystem called NHLBI BioData Catalyst. Seven Bridges developed a platform that's part of this BioData Catalyst ecosystem. I'm going to start off by going through some slides, and then Dave is going to do a demo of the platform after that and show you what some of the things I talk about look like on the platform. Let's start with why BioData Catalyst was created in the first place. Over the last 10 years, researchers have continued to improve genomic sequencing methods, making it faster and cheaper to generate genomics data. And this has led to numerous data sequencing projects funded by the National Institutes of Health and the creation of a wealth of large genomic data sets that can fuel research in precision medicine. However, these increasingly large data sets bring a number of challenges to data analysis. Some universities are able to offer their researchers access to high-performance computing infrastructure. In these cases, storing and managing the large data sets is possible. However, institutional agreements need to be put in place to ensure that these sensitive, controlled data sets are handled appropriately. It's also really cumbersome to store and manage petabytes of data, and it requires resources to manage the data and ensure that the latest versions are available. To perform analysis, researchers must wait their turn in line. And it could take weeks to process thousands of samples when you might want the results tomorrow. For researchers who are working on collaborative projects, there aren't really easy mechanisms to share results and methods with other researchers. And then finally, and perhaps most importantly, it's important to note many researchers are based at universities that don't have this high performance computing cluster that would be necessary for managing and analyzing large data sets, preventing them from utilizing these resources in the first place. So the National Heart, Lung, and Blood Institute recognizes these challenges, and several years ago, they decided to build a cloud-based platform to democratize access to data and computational power. This platform is called NHLBI BioData Catalyst, and it enables researchers to access heart, lung, blood, and sleep data sets. And this includes a combination of genomics data sets, clinical data, and imaging data. These data sets are all stored on the cloud by NHLBI, so researchers don't have to worry about managing the data. And the platform also provides tools and access to cloud computing infrastructure of Amazon Web Services and Google Cloud, so that researchers anywhere can access and analyze these heart, lung, blood, and sleep data sets. BioData Catalyst also democratizes access to large data sets. So researchers from across the country can log into BioData Catalyst on the web and access the same copy of these data sets, eliminating the need to manage the large data sets locally. NHLBI supports the cloud storage costs of hosting these data in AWS and Google Cloud buckets. So that's what's kind of depicted over here. And then also importantly, the BioData Catalyst ecosystem has an authorization service that manages user permissions on the data programmatically, such that when users log in here to a BioData Catalyst platform, they can only access files in the AWS and Google Cloud storage buckets that they have dbGaP approvals for. Currently, there are 3.4 petabytes of data available in BioData Catalyst. There are over 80 studies from the Transomics for Precision Medicine Initiative, or TOPMed. These offer a wealth of phenotype data that's related to heart, lung, blood, and sleep disorders. Some of the studies have thousands of phenotype variables measured, and you can access both these raw phenotypes that would be available for download on dbGaP, as well as harmonized phenotypes that researchers have created directly on the system. As for the genomics data, BioData Catalyst currently hosts the aligned reads. This is in the form of CRAM files, as well as the variant calls in the form of multi-sample VCF files. Other omics data, including metabolomics, transcriptomics, proteomics, those are going to be added to BioData Catalyst after they're released on dbGaP. So that's kind of the normal flow for making data available on BioData Catalyst right now. This combination of the phenotype data and the genotype data in TOPMed makes it a really powerful resource for association testing. So we have a lot of researchers doing that kind of analysis on the system. If you're interested in using these data sets, you would just work with your PI to get access in dbGaP. The system also hosts 1,000 genomes project data. This is an open access data set, so it's really valuable for just testing out the system, learning how to use it. Okay. So how do you find these data sets and work with them? BioData Catalyst is an ecosystem of cloud-based platforms and services that are integrated with each other. There are a couple of search services where the researchers can search through the hosted TOPMed phenotypic variable data, and then they could form a cohort of subjects of interest. And then once they've formed their cohort, they can send that cohort to a workspace environment, choosing between 7Bridges and Terra, and there they can run analyses at scale and make use of hundreds of tools and workflows that are directly available on 7Bridges, or they could bring in reusable workflows from the DocStore tool repository. All of this sits on top of the infrastructure for managing the hosted data sets that are stored in those NHLBI cloud buckets, because NHLBI is supporting the cloud cost that way, and then the user permissions being handled programmatically. So for the rest of the talk, I'm going to focus on what you can do in the 7Bridges cloud-based workspace environment. 7Bridges provides private, secure workspaces for running analyses at scale. Within the workspace environment, you can access and use the 3.4 petabytes of hosted heart, lung, blood, and sleep data. For those who are conducting collaborative research projects, you can easily collaborate with other researchers in shared workspaces. I'll show you what that looks like. And to perform analysis, you can use hundreds of pre-configured, optimized tools for the cloud, including association testing, RNA-seq, methylation, annotation, and more. And Dave's going to show you a couple of these in the demo. Using these tools, you can analyze thousands of samples at once and really take advantage of the power of the cloud. You can also work interactively, create plots for your paper. So essentially, you can run your analysis and create figures for your paper without leaving the platform. As a workspace environment, 7Bridges enables users to organize their files, methods, and results into what we call projects. These are also called workspaces or sandboxes, depending on the type of environment you're in. Users can create as many projects as they like on the platform and then add in their files for analysis. To set up and perform an analysis, the user would also bring in their app. This is our term on the platform for bioinformatics tool or workflow. Using the user interface, the user can configure the app, select the input files, click run, and then the results get organized in the project. You can also run things via the API if that's your jam. Users can also analyze their own data on BioData Catalyst. There are several options for conveniently bringing your own data to the platform. There's a command line uploader, desktop uploader, API, FTP, HTTP import. You can drag and drop files directly from your computer. So depending on your use case, lots of available options. If your data is already in an AWS or Google Cloud bucket, you can just connect that directly to the platform and avoid moving the data entirely, which is pretty cool. So this enables you to treat your private cloud storage as an external file repository to projects on the platform. And then once a secure connection is established, these get treated as any other file repository, and the users retain full control over access, management, and integrations. To enable researchers to work with data that's stored on distributed cloud locations like AWS versus Google, the platform lets you compute on both AWS and Google Cloud from the one user interface. Users get to select whether they want to run their computation on AWS US East 1 or Google US West 1. These are the two available cloud locations right now. This list will expand in the future based on user needs. So if you have your data in a private AWS bucket, then you can perform the computation on AWS and avoid any data egress over to Google. So that just makes it as efficient and cost-effective as possible. If you plan to work with the hosted data on BioData Catalyst, like the TopMed data, you can find these data sets directly on the platform. There's a feature called Data Browser. And you can see here in the Data Browser, you see the list of all the hosted studies. You can select a study, bring it over to this graphical query interface, and you could find a set of files there. So the user could find all of the multi-sample VCF files for a given study. And then once they have the files they want to use, they click Copy Files to Project. This links the data to the project, but you're using that copy of the TopMed data that NHLBI has stored in the cloud bucket. You're not making a new copy of the data. And then importantly, this is all based on your permissions. So if you don't actually have approval in dbGaP to use that TopMed study, you're not going to be able to copy it to your project and use it in an analysis. Within your project, you can manage files in this Files tab that's shown here. You can make folders to organize your files. So we've got 4 folders here, some for raw phenotypes, another for harmonized phenotypes. You can filter your files based on file metadata. So we're showing a couple of categories here. You can toggle and add a bunch more under this menu. All users have the option to work alone in a private project. So in this one shown here, you look at the members box, I'm the only one there. So this is a private project just for me. If I upload data to this project, I'm the only person that can see and use that data. So it remains accessible only to me. But let's say you're working with collaborators, whether it's someone in your lab or someone at a different institution. Platform makes it really easy to collaborate with those individuals in the same project. So the project owner can add collaborators as members, and that happens in this box here. So we've got 4 individuals working together right now. If we wanted to add someone new, we'd click Manage Members. We're adding Sarah, and we can set really granular permissions on what Sarah can see and do. So right now she has write, copy, and execute privileges. So she can add new files to the project. She can download data, and she can run analyses. If you want to minimize that, you could give her read-only access. There's a lot of flexibility there. So now let's move from data to tools. A great place to start is our public apps gallery that has a curated collection of over 500 bioinformatics tools and workflows that have been optimized for speed and cost in the cloud by the 7 Bridges bioinformatics team. These tools are fully parameterized and they're customizable. You can run them from the user interface or use the API. And you can learn more about the tools by clicking on these cards that are shown here. And then you can see a full description of the tool, troubleshooting tips, required input files, expected outputs. So they're fully documented tools in this public apps gallery. Here we're searching GATK, so you can see the offering there for those best practice workflows. All of the available tools and workflows use a combination of Docker and the Common Workflow Language, or CWL, to enable portability, reproducibility, and scalability. The Common Workflow Language is a community standard workflow description that describes all of the requirements a tool needs to run in a reproducible manner. So that's how it lends itself well to reproducibility. The platform features a web editor. That's what you see here with all these nodes moving around. You can use this interactive interface to combine CWL tools into longer workflows. You can also port in CWL workflows from external repositories like DocStore. And finally, you can create your own CWL tools using our CWL Web Composer. You're welcome to learn the syntax of CWL if you're interested, but the platform makes it so that you don't have to do this. With the software development kit, you can easily create tools, chain them together into workflows without any programming experience, and the software development kit is going to create the CWL for you. So what this looks like for the user is they bring in their script, they bring in a Docker image for the tool, and then they're adding the CWL with the software development kit. When you're running CWL tools and workflows on the platform, you can scale to running hundreds, thousands of analyses. We call these tasks, all in parallel. We call this batching. You could batch with by file or file metadata. And so essentially you're just kicking off a number of tasks in parallel, processing them with the same workflow. So this is really great way for running analyses when you're doing like whole genome sequencing kinds of analyses or whole exome sequencing. And you could just batch by sample and do your variant calling pretty quickly. University of Alabama professor Zhe-Chen Chang took advantage of BioData Catalyst and batch processing to advance his project goal of systematically and comprehensively characterizing structural variants and their contributions to phenotypes in 2 TOPMed studies. The platform enabled him to get direct access to thousands of whole genome sequencing samples because the TOPMed studies are hosted by NHLBI in the cloud. So that's what he was able to run his analyses on. So he didn't need to deal with, downloading the data to local server where the disk space might be constrained. And then he was able to easily deploy and distribute software packages to the cloud. And finally, dramatically reduce the time for data analysis from months to days. So he ran a structural variant calling pipeline on nearly 3,000 whole genome sequencing samples in 1 day, which is pretty cool. For users who need to perform phenotype harmonization or fast prototyping of custom tertiary analysis tools using interactive Java, Python, and R, you can work in the platform's Jupyter Lab notebook or our studio environment. There are over 50 instance types to select from. We encourage you to try out BioData Catalyst and create a platform account. You're going to need an ERA Commons ID to do so. If you don't have an ERA Commons or you don't know what that is, I recommend talking to your PI. They'll know who to contact. To help you test out BioData Catalyst, NHLBI generously provides $500 in cloud credits to each new user. So when you create your account, you're automatically going to have this $500 in credits added. So then you can just get started with testing. And if you decide to perform a research project, there are ways of requesting additional credits from NHLBI, and you can learn more on their website, biodatacatalyst.nhlbi.nih.gov. The platform is really well documented. There's lots of tutorials, so I recommend checking these out as you get started. We have a 24/7 support team that you can contact directly through the platform by clicking on this question mark, and they will respond within 24 hours, often within a few hours, so they can help you with troubleshooting analyses. Dave's going to do his live demo next, but if you'd like to see, and if you like what you see, you want to try the platform yourself, we encourage you to join us for a 1.5-hour training. During this training, you'll receive step-by-step guidance on how to use the platform to create projects, manage files, and run a workflow. So I ask that you fill out a Google form that I'm going to put in the chat when I stop talking, and indicate your interest by end of day Tuesday. And I'll ask for your name and email and all that, and then we'll contact you and try to set up a group training. You will need that ERA Commons ID, so please start looking into that if you're interested. And now we're going to transition over to Dave. We'll do questions at the end.
Thank you so much, Alison. Oh, that was a great introduction and overview. So the next part of the presentation will be a live demonstration, the objective of which is to learn. I want everyone to get a feel and learn about the capabilities and feature sets of BioData Catalyst powered by 7Bridges and what that looks like live. And to do that, we're going to first frame briefly a scientific question. Then we'll introduce a user flow model. So this is a typical user progression about how they click through the platform to do their analysis. Sit back, I'll do a demo. You don't have to do anything. And then, of course, we'll have time for questions. So the questions can be about anything in the demonstration or anything that Alison went over. The scientific question we're going to frame our demonstration on is about cardiac ventricular failure. And so there was a publication in the Journal of Clinical Investigation that this group actually came up with a way to predict right ventricular failure by finding that this particular gene pathway is actually a conserved mediator of that. So the idea is that they could analyze the heart tissue, the gene expression of the heart tissue, to determine if right ventricular failure was imminent in these patients. So this is a really great paper. What we're going to do in the demonstration is use the raw sequencing reads from this paper that was put on the SRA, short read archive, and that's a framing. So we're going to do a similar analysis as was published in this paper. Next slide, please. So no matter what you're doing on the platform, there's a typical user flow, and that Alison went over most of this, but it involves 4 categories: project creation, staging input data, workflow staging and potentially tailoring workflows to your needs, and then finally running your analysis. So we're going to go through this in the demonstration. I'll create a project for today's specific analysis, and then we'll stage some data. So we're going to start with reference files. So that's always sometimes difficult to find the correct reference files. 7Bridges hosts reference files for many different types of analysis. So we'll just link that to our project. And then we're going to ingest the heart gene expression data from the short read archive. Next, we're going to add tools from the public apps gallery that Alison already mentioned. So we're going to have tools to do RNA-seq analysis inside our project. one of which we're going to tailor to make it a little more unique for our analysis. Finally, we're going to run analysis at scale. So we want to show off the ability to use multiple nodes. So we're going to use a batch task and scale across multiple cloud nodes and do some of the analysis in parallel. Next slide, please. So we're going to focus today on common workflow language workflows that Alison talked about. So 3 in particular. So we'll grab the short read archive downloader and set metadata workflow from the public apps gallery. And this will allow us to automatically download and set metadata from SRA. Then we'll run trim galore on the raw sequencing reads. And what this does, if you're aware of next generation sequencing, this is going to trim back the low quality reads at the end. And it's actually going to give us a FastQC report. So it's two parts built into trim galore there. And finally, we're going to run a really complex workflow that's going to go from RNA alignment to quantification, differential expression, and finally pathway analysis. And we want to see if any genes are upregulated or downregulated in the different conditions. So we'll do biventricular heart failure as well as left and right heart failure. Okay, next slide. So this, Alison already talked about this, but inside the projects, there is a feature called Web Composer. And this is what the canvas looks like. You actually can connect multiple analysis steps together using this graphical interface. And we're going to pull 2 apps from the public apps gallery, HiSat2 into string type quantification. Then we're going to pipe that into differential expression, and we're going to add a custom sub workflow. So workflows can actually contain steps that have multiple steps in them. So you have this nested ability, and we're going to pull in a gene set enrichment analysis workflow, and I'll show you how to tailor that. So I'm going to get started with the demonstration. So I'm going to go ahead and share my screen. So I'm going to share my entire screen, and let me just pull this window over here, and you should be able to see the platform now. Okay, so I'm already logged into the platform. So Alison mentioned that you'll need your ERA Commons ID to log into the platform. I've already done that. And this is what the platform dashboard looks like. And it's what you'll see is your most recent projects on the left side. Okay, and I belong into a bunch of different projects. And then you'll see your most recent analysis on the right side. And analysis is actually 2 different tabs. So today we're going to focus on common workflow language tasks. So cloud batch jobs, and those will be under the tasks tab. And this is an overview, and you'll see one actually had almost 2,000 jobs here. But you can, we'll go over that later. But there's also another way to do analysis interactively, which Alison mentioned, and currently we can run Jupyter Lab, RStudio, and SAS. So these are private environments that you can spin up on a single node and do your prototyping and interactive data analysis. So let's go ahead and progress along our user flow. So the first thing we need to do is make a project. And I'm going to do that by clicking the projects tab and then clicking the create a project button. And here, first thing we're going to do is name the project with our purpose today, which is the RNA-seq analysis of heart failure. Okay, and then this actually gives us a custom URL, which you can bookmark and to access quickly later. So 7 Bridges has billing groups to keep track of your cloud funds and what you're spending on your different projects. You can belong to multiple billing groups, and Alison went over that a little bit. Today, I'm going to use the training billing groups, what I use for these types of webinars. But just know the billing groups come with the platform, and they help you keep your storage and your analysis cost organized. All right. And then Alison mentioned that we are a multi-cloud platform, so you can choose which project you want this, the derived data to either go to AWS buckets or Google buckets, and then the computation would follow that, right? So either AWS or Google. We're going to choose AWS today because one of the template projects I'm using already has data on AWS. So I want to make sure that's the same. So when we can transfer data better that way. There are a couple other execution settings. You can use preemptible instances, and that's very helpful. You can also reuse pre-computed results inside your project. So we call that memoization. The final option before we create this project is we need to indicate if this project will contain the NHLBI hosted TopMed data. And if it does, you make a controlled project. And what that does is it adds additional guardrails to make sure we're using that data appropriately. But today we're going to do a bring your own data situation. So at that, we don't need that extra programmatic control in that case. So after we fill out this project wizard, we hit create, and it immediately sends us to my private project. So as Alison mentioned, when you make a project, you're the only user that can see what's going on in this project. And let me just orientate you to the project. You have the title in the middle, and on the left side, you have dashboard, files, apps, and tasks. And so this mirrors the user experience flow that we want to emulate. So we're starting in the dashboard, then we'll go to linking files, linking apps, and then running analysis. Okay, so on the left side, we have a description. So this comes with your new project, but we can always edit this description and take notes about what we're going to do. So I'm going to do that right now. I'm going to go switch to my template project, and I'm going to copy these notes. And I'm going to add them to our new demo project so we can remember, I can remember what I need to do. So we're going to look at the RNA-seq analysis of heart failure. And here's our analysis steps. And then I have some links, helpful links here. So that's the description. On the right side is your project membership. So you can invite, you can analyze data privately, or you can invite collaborators. So I'm going to demonstrate how to invite a collaborator. So you just type in either their email address or part of their user ID. And so I'm inviting Alison. And at this point, you could specify their permission level. So you can give them read only or some combination of write, copy, execute. If you give them admin, that they inherit all the permissions, and then they can be a co-admin of the project, which means they can invite other users and change these types of settings. Okay, so I'll invite Alison, and you see that we have two members now of the project, Dave and Alison. Okay, the final thing on the dashboard is the analysis window pane here. So this is just a quick overview of the tasks that we're going to run in the project. So after we fill out the dashboard, the next thing to do is add files. Okay, so files are the basis of every analysis. So let's go ahead and link some files. So there's multiple ways to bring data to the platform, and here are some of the examples. The first thing we're going to do is link public reference files. So this is a really handy feature of 7Bridges. We have hosted reference human genomes and annotation files, so you don't need to go searching for them, and they'll actually link to your project, and they're hosted on 7Bridges buckets. So I am actually going to flip back to here because I want this note. I want it's a very specific reference I'm looking for. This is the GTF file. So all I do here is I'm in the public files tab, and I just search for the name. And you'll see these are anything that has to do with this particular GTF file. I'm going to go ahead and select that one. And I'm going to, this is the actual genome archive, which includes the genome, the transcriptome, and the index. So I'm going to paste that in. And so I've selected two files. and I'm going to copy these to my project. At this point, I can tag them as references. So that's a really handy feature to keep things organized. And they immediately get linked to your project. So I have these 2 files and they're tagged as reference. Okay, there's the next thing I want to do is actually, we're going to be importing from SRA. So I have the link to the SRA accession number. I'm going to open this up in a new tab. So this is the new to me NCBI SRA run selector. And it actually says that SRA data is now in the cloud, which should give us faster transferring, as well as they have a revamped, a really nice web page here. So this accession number comes directly from the publication I was talking about. And the only thing we need here to get from this web page is the metadata, file metadata text file. So I'm just going to download that to my desktop. And it's called SRA runtable.txt. I'm going to flip back to the project and I'm going to add files a different way. So I'm going to add files directly from my computer. So this is going to upload through the web browser. So it says drag file. So I'm just going to browse for this file on my desktop. And this is a really handy way to upload manifests like this, documents or scripts. This is really, really quickly uploads. Okay, so now we have 3 files. So we have the 2 references and the SRA run table text file. By the way, I'll mention that files are previewable, and I'll show this a bunch more, but if you just want to get a preview of what your manifest looks like, you can just click on it and you see that it has all this information. It's very handy. One more way before we talk about software, there's one more way I want to highlight to link data. So we're going to link data from a previous project of mine. And because we are limited in time in this demonstration, I'm going to do a lot of cooking show style demonstrations where I'll start a task and then I'll flip to the results. So I've already computed the results in another project. So I'm just going to grab those for us. But this also shows you how you can link from other projects. So I have a template project and I have a results folder. I'm going to copy that to our project here and I'll show you what's in it. So these are the cooking show results. So I have results for SRA download and the next step, which is trim galore. So that's it. So here's what our files tab looks like. We have 2 references, an SRA manifest, and the results cooking show folder. So the next thing is to keep moving along. So we'll add some apps. And Alison mentioned that we have 500 plus publicly available, optimized common workflow language tools. And you can add them by clicking Add App. And then this automatically takes you to the public apps gallery. And you can search by category. So we have GWAS and copy number variation, DNA methylation apps. So those are all there. You can also search by toolkit. I know the exact name of the tool I want, so I'm just going to type in SRA, and that automatically filters anything to do with SRA. And the first one is the download and set metadata app. So I'm going to go ahead and copy that. Okay, and there's another app I want to grab, which is called Trim Galore. And this is actually a wrapping around Trim Galore and FastQC. So this is really handy for not only trimming your reads, but also doing a quick quality control check. So those are the 2. Okay, so I grabbed those 2. I want to grab one more, but I'm going to link it from my template project. So you notice I was in the public apps tab. You can also link tools from other projects. So I have the template project, and I want to go ahead and link the tool that I started to create for RNA-seq quantification all the way through pathway analysis. Okay, so let me X out of here. Let's see what we have. So we have SRA, download, trim galore, and RNA-seq alignment. So let's go ahead and tailor a workflow, and then we'll move on to running some of these workflows. So if you click on any of the workflows, it'll give you a preview of what we have here. And then you click edit. This will immediately take you to the web composer. Okay, so this is the web composer that Alison mentioned, and you can move these tools around. You can drag new tools from the left side onto your workflow canvas and connect them. So you have all the 500 plus public apps that you can start with, and that's how I grabbed HiSat2, StringTie, and DESeq. These are public apps, but I'm actually wanting to add a tool I was working on that I wrapped myself called Gene Set Enrichment Analysis. So it's another open
Join
source.
the meeting.
Yes. I was clicking too fast. Okay, so I have this workflow called Gene Set Enrichment, and I'm going to drag this onto our canvas. Let me zoom in for you so you get a better look. So what we want to do is the output of the differential expression analysis. We want to pipe the DESeq2 results directly into this workflow, and this will give us a nice enrichment report that we can look at later. Okay, but at the same time, I want to save these DESeq2 analysis results because those are really handy to look at when you're doing this type of hypothesis testing. In addition, DESeq2 makes a really nice HTML report, which we're going to save. Okay, if you ever need to hard code some parameters in your workflow, you can just simply click on an app, and I'll specify the gene set to use for the gene set enrichment. Also, you can easily find the information about apps. So here is the HiSat2 StringTie workflow. It gives you the description, but it also tells you how much it will cost to run at the bottom here. So I can expand this and it'll show me if I'm doing 400 samples or 100 samples, this is the expected duration, the expected costs, etc. So that's very, very handy as well. So that's all I needed to do to tailor. We just added that one step at the end, and I'm going to go ahead and save. And I can add a revision note. So this is really great for the developers and the audience. There's always revision commit notes, and you can go back in time to previous versions. Added gene set enrichment analysis workflow. All right, so let's go ahead and run some analysis. Okay, so we're back in the apps tab. The first thing we want to do is download the SRA data. Okay, so we have this great tool called SRA download and set metadata. So I'll go ahead and run that. And when you run an app, it immediately takes you to the draft form you fill out. And so the only input here is the metadata file, which I uploaded. Okay, and it automatically knows to look for that type of file. And we also need to specify the accession number, which I'm just going to grab from here. I'm going to just paste it in, and I'm going to hit run. Okay, and as I mentioned, this is a cooking show, so we're going to compress time. This will take, I just timed it earlier, it's 24 minutes to download all the 30 FASTQ files. But let's go ahead and see what the output would look like. So in the results folder I have the SRA download. And what I want to show you here is this is a really great view of not only the file name, but you have the type, but you also have this metadata field. So these metadatas are automatically populated with this tool. So it's grabbing it from SRA. And this platform allows you to attach metadata to files. And then as you use those files in the analysis, the metadata is inherited, right? So that's a really unique thing. So if we trim or manipulate these files, we'll still know that this file came from the right, this sample came from the right ventricle, and there was no heart failure in the subject at that time. And you also have the sample name, etc. So we're going to use these in our analysis because we want to focus on the condition. So we want to focus on, was there heart failure? Was it left ventricle heart failure? Or was it biventricle heart failure? Okay, so let's run the second app, which is the Trim Galore. So we find Trim Galore in our apps and we hit run. And by the way, you can do this from command line if you're more of a command line person. Alison mentioned there's an API, but I prefer to use this. graphical interface. So for here, the only thing we need to fill out is the input reads. So input reads are fast queues. And the app knows that only fast queues can go into this input port. And so we'll just go ahead and select all 30 samples. We'll save the selection. And now here's what I want to show you. We can batch to make this finish faster. So we can batch by either file, which I'll do, or file metadata. So sometimes you want to, so what this will do is send each of these files to its own Amazon Cloud node. So we're going to spin up 30 Amazon Cloud nodes simultaneously. Sometimes you want to send all files that belong to a particular subject to one instance. And in that case, you would choose file metadata, so you could batch by subject ID, etc. So this is saying that there's 30 groups with only one file in each group. So this is not a paired end run. They didn't do paired end in this study for some reason. So let's go ahead and run. So I click run, and the platform warns you. It says you're about to run 30 child tasks. That's okay, because we want to scale horizontally. So let me just refresh the browser. Okay, and we have these tasks are spinning right up. So just another refresh, and you'll see that we have all 30 tasks running at the same time. All right, so once again, cooking show, I've already created the results. So those are in trim galore. And these are, I just want to show you that the output will be another FASTQ file with underscore trimmed. So we know it's trimmed, and the files have also inherited all the metadata we need for the next step. So that's very important. So we don't need to have another manifest. We can just use the files to create their own manifest. So we'll take questions about that later, because it's a really important feature of the platform. So let's go ahead and run the last app, and then we can take questions. So the last app is the RNA-seq quantification, alignment quantification, differential expression, and pathway analysis. So it does a lot of things, one right after the other. And this takes about an hour and a half to run. But cooking show, I've already run this, and I can show you what the completed results look like. Let's go ahead and kick this off. So we first select the reads. So we need to be careful, because I only want to use the trimmed read. So I could simply just search for trim, and it'll match part of the file name really quickly. So I can just pick all 30 of those. And we're not going to batch these. I want DE-seq needs them all together to do the differential expression. So we're going to not batch. We're going to use a single node, although it does do a lot of parallelization. So if there's time, I can show you that. We need the annotation file. It knows what that is. We need a reference, which happens to be the tar of the transcriptome reference. That includes the indices. And then we need to specify the covariate of interest, which is the condition column. So let me show you what it looks like on one of the files. So I open up one of the files, and you can see all the metadata that's filled out. So the covariate of interest is this field called condition. So this subject was not having heart failure. That's the control or the reference. So I'll paste that in. And the other one, let me see if I can grab it. I just want to make sure I spell it correctly. Which one did I get lucky here? No, that was not failing. So let me go back to the files and get the results here for trim galore. And I want to use biventricular heart failure. So I just want to make sure I spelled that right. Okay. And then I click run. And that will go ahead. And so let me just show you, we have a few minutes left. Back in the template project, I ran this earlier today. And this actually gives you a tremendous number of outputs. So you have input tables for ball gown. You have the DE-seq2 analysis results. So by the way, anytime you see a histogram here, that means you can view the results right on the platform. So we have all these count matrices. We have the gene enrichment report, which is basically a web page that's been zipped up. But we also have a really nice HTML report that was created by DESeq, which I can expand here for you. So all HTML, PDF, and images are viewable right in your web browser. So these links will work. So I can jump directly to PCA, and I can see, clustering that's clustering pretty good with not failing together, and then biventricular heart failure down here in red, and left ventricular heart failure up here in green. You get a cluster dendrogram, as well as normalization checks, etc. So
Dave,
that
we
said,
only have a few minutes left.
Yep.
Is this an okay place to stop?
Yep,
So we
that's
can
all
take
I
a
needed
couple
to show.
questions? Okay.
Yeah. Should I stop my screen, or let's take some questions, I guess.
Yeah, so someone asked in the chat, if they want to use a custom tool, does it have to be in CWL? Could you run SnakeMake or Nextflow?
Great question. Okay, so CWL just has to be the outer layer. Inside that layer, you can actually run whatever you want. So I know a lot of users are running SnakeMake inside the tool.
Okay.
Now, I haven't come across the Nextflow, but I think we could make that happen.
Yeah, so come to the training, and then we can talk a little bit more about what kind of tools you want to run.
Yes.
Yeah, and another question is, can user customize Python script too?
Yes.
I think that's a yes.
Yeah, absolutely. And there's a great way to do that. We do a whole training called Bring Your Own Tools. And one of the primary use cases is users have a Python script or a Python notebook. So you can actually use that as input into an app, and it'll run the app. You just have to make sure that the Docker contains all the libraries.
Yeah, and another thing I think some bridges is good at is to optimize your pipeline. So if, for example, I have a pipeline no break that was deployed to the, I think this BD Catalyst also have that pipeline.
Yes, I think so.
Yeah, so it can optimize your pipeline and make it faster and use less resources. So that's pretty wonderful.
Absolutely. And I do, when I'm wrapping tools, I use this screen a lot. So this is drilling down into a sort of a micro level of what's happening. So you can actually see each of the steps in your workflow and how long they're taking and how much parallelization you can do. So here, string tie is actually doing a high level parallelization. Each line represents a job. And then you can actually look at the instance metric. So this is what Zaichen was referring to. You can see that we've optimized the CPU, but the RAM is not really using all the RAM that it had. So you could maybe dial back on your RAM allocation there.
Yeah, there's another question. I think, can we log in and navigate through SSH and then open through IDE like VS Code?
So there's a 7 Bridges command line interface, which allows you to do some of the functionality from a command line. You cannot SSH into a running common workflow language tool, but you can spin up a Jupyter Lab or RStudio environment, and you do have terminal access there. So, and you have sudo in that particular instance, so you can prototype things in that environment.
And I think when you have your own data, probably you need to use the SSH command to access or upload or download the data to or from this cloud platform. I have been using that for a while, so yeah. I think that's a yes, kind of a yes. Yeah.
Yeah, so there's the command line uploader, the command line interface, and then there's also a system called SB Files system, which allows you to mount your project to various different environments.
Yeah. How about IDE? I think that's the interesting question.
So great question. It depends if you consider RStudio or Jupyter Lab and IDE. So Jupyter Lab is not just a notebook, it's terminal, notebooks, etc. So you can start those up and they'll have inside your project, you'll have access to all the files.
Okay, any other questions? Okay, thanks,
Good question.
Alison and Dave. Great talk and a great demo. I think that's really interesting, and I think I would you mind I share this form to the community, to the I have emailed.
Absolutely, yeah, because maybe people will watch the recording after and then decide they want to join the training. Totally fine.
Thank you so much.
Yeah, thanks for having us.
Yeah, I saw Laura and Wes is here. Hi, Laura and Wes.
Hey, how's it
How are
going?
you?
Yeah, I'm good. Glad to see you here.
Likewise.
So I will stop.
I'm gonna stop. Thank you.
Okay. Thank you. And Laura, probably you can share your screen. Always.
Yeah, I can
Yeah, Laura's
share.
going to share. Yeah.
Okay. Thank you.
Okay. Can you guys see the slides?
Yes, we can.
Yes.
Okay, good. So I guess, yeah, we'll just go ahead and get started. I'm Laura. I'm a PhD candidate in the computational biology program at Anschutz Medical Campus. And I was one of the first beta testers that came onto the precision medicine platform. And now I am a lead data scientist at the American Heart Association, where I focus pretty heavily on the strategic development of the precision medicine platform and just developing and optimizing the platform for research analytics. And then I also do, I guess, conduct my own research using the platform, just doing mostly data integration and data mining for clinical risk assessment. And then Wes.
Hi, everybody. I'm Wes Wiggins. I'm the delivery lead from Hitachi Vantara. We've been building and supporting the precision medicine platform since its inception. And I've been involved for about a little over 3 years now. So yeah, my team is the one that actually supports the production end users, researchers, but also builds out new features and enhancements in the PNP. And I'm happy to be here. Nice to see you all.
So I think I just wanted to, quickly, like, I'm sure that the last talk was structured in a similar manner, but please feel free to put questions in the chat or just kind of interrupt at any point. If there's, lots of questions, that's a good thing. It'll help us drive, the content that should be delivered. And then also, if there are things that are in our presentation that is, somewhat repetitive to the BioData Catalyst presentation, Zetchen, please feel free to just tell us to skip a slide or anything like that. So I think, I guess with respect to cloud-based research, exactly, hold on. Okay, there we go. With respect to cloud-based research, like in the precision medicine platform, with, I guess, in general, the future of cloud, I think one of the main things that why the industry and also the academic fields are heading towards cloud-based computing is that it helps in the data access and governance, as well as access to software, especially licensed software and use. And it facilitates that, streamlines that. It also reduces the cost of resources, especially hardware, and kind of facilitates and enables reproducibility and reuse of analytics that may not have been really possible before. And so this is definitely true with respect to environments that we set up. So cloud-based environments, especially within the platform, when you're accessing an environment that has the same operating system and is consistent with respect to version and software, even if it's just within a project, that really facilitates users not having to deal with issues of, one person on the project has R and Python set up differently on their computer than another computer. And then it also facilitates the ability for us to reproduce projects across projects, such as just being able to download an environment or run a project in the same environment that was previously set up for that analysis. And it also allows us to facilitate access to software. So I'm going to kind of demonstrate that a little bit in our demo. And then I think the other thing that really the cloud-based computing that provides unique, I guess, benefits to is that the it provides an access to high performance computing, but it provides an access to dynamic and diverse options. So many of you might have at your university like a cluster or a certain high performance setup that then you can access via a queue or access via Slurm or some managing software that then allows you to access that. And so then the university is typically investing in hardware and the way that it's managed on a periodic basis, basically just to update the software and the hardware so that it's the most recent. But then also it's kind of set in stone to a specific setup that caters to the majority of cases. Whereas with the cloud computing, you have the ability to really dynamically set that up. So if you need a certain specific setup with GPUs or CPUs and a certain number of CPUs, certain amount of memory, all of that is dynamically scalable and available, basically just by clicking a button. And then additionally, by allowing users to access similar environments or shared environments, it really secures the ability facilitates the ability for users to collaborate and streamline their collaboration and development on a project. And it also allows for us to have more securely, I guess, shared data, especially protected data that's related to either human subjects or just any kind of protected data, really. So I think the general approach that the NIH, the AHA, and definitely we're seeing this more and more over institutions. So at my institution, we've just kind of set up something like this for our medical record data as well, where the data is actually housed at the institution itself. It stays there, or at the NIH, the organization itself, so the NIH or the AHA. And then the data access is delivered securely to a workspace. So the workspace is actually coming to the data, and you can access the data in a FedRAMP HIPAA secure environment that then is already equipped with analysis and statistical software that's needed, and facilitates the, I guess, computational resources that would be needed for more intensive analyses. And so I think the ideal or the reason for this approach is that it helps improve definitely the cost that institutions have to maintain with respect to hardware or software licensing, and also is more efficient. And so to give you an example of some of these things, especially with the AHA that we're working on right now, we have a partnership with BioData Catalyst, and we're in a pilot with the NIH, a variety of different departments, the BioLink, for example, and NHLBI, and then also like the Cancer Institute. And where the idea is that you would be able to search across these different portals or make requests for different data sets. So for example, if you wanted to request access to the Framingham data or access to data that you just saw that was available in BioData Catalyst, then you would be able to access that in a workspace in multiple environments, and those environments would be interoperable and reproducible. So if you were to pull data, let's say from the NIH into the precision medicine platform, you'd be able to do analyses that way in that workspace, or you'd be able to do them in the BioData Catalyst platforms that are available, such as 7 Bridges, or I think it's Anvil, that is their other one that they currently have supported, and then we're also on that list. And so then the data will all be just housed and maintained at the various institutions that own them, but your workspaces will be able to access whichever data you need. So I think I wanted to just, kind of address some of the challenges or maybe growing pains that are associated with kind of cloud computing, because it definitely can, in some ways it is like a great solution and a great advancement in research and analytics and just computing in general. But we are new, and so it's new and it's different, so that can sometimes have a little bit of a learning curve. As you saw with I'm sure with BioData Catalyst, the way that you run and perform analyses is quite a bit different than you would set it up on your personal computer. You might not have the same IDE that you're using, or use the software or interact with it in the same way. Or same is true with, you're not going to be interacting with the high performance computing options in the same way. It might be easier, but it still is just new and different, right? The same thing is true for using the data within a secure environment. So with respect to from, I guess, like a perspective of a data distributor like the NIH or the AHA or a hospital institution, the hospital has more confidence that the data is being used in the manner that it was intended. Also that it's following the appropriate security protocols, and there's not necessarily a risk, less risk that's associated with just delivering copies of the data set to multiple users on their own computers. And also it helps control versions of the data that are being used. For example, just always having the latest version of the data available. But one of that is there are limited, I guess, components because of that security. So one of the things I think that I heard at the end of the BioData Catalyst call, BioData Catalyst, was the ability to SSH in and use your own IDE, for example. That's kind of common, I think, for certain supercomputer setups at institutions and universities. Whereas typically the cloud, like a cloud-based platform for security reasons, would not necessarily facilitate that. But we have gotten other ways around that where you can open up your own IDEs in certain environments, that kind of thing. So it sometimes can be a little bit, I guess, and also getting data in and out sometimes requires a secure protocol. So it's just a little bit more restricted than maybe what you're used to. So I think sometimes that can be frustrating, which is another reason why we need your feedback as users so that we can make those as usable and as user-friendly as possible while still meeting all the standards of that FedRAMP or the other security protocols put in place. And that kind of just brings me to the next piece, that the platforms are still being designed. So for example, I think with BioData Catalyst, they've really solidified a lot of the way you search and access data within their workspace. And that's, I think, pretty cool, the way that they've got that set up. I think their platforms that they have currently have been really a little bit more focused on genetics. So they chose to start there and are kind of expanding to image analyses or other analyses. I think whereas the precision medicine platform, we're a little bit less, I guess, not at the same point along as being able to search and request data sets or a variety of different data sets, just because we don't have as many data sets that are available or that are owned and maintained by the AHA, maybe managed by the AHA. But we do have, our platform is designed for a little bit more dynamic types of analyses. So we have a lot of image analysis and genetics that are available in our workspace. So we're still, I think all the platforms are still kind of in this phase of developing based on different use cases and catering to those. So again, we need your help to identify which use cases to cater to, right? And which ones are most important and which ones are, which cases, certain cases that don't get covered, that kind of thing. One of the, I think was Zechen for sure, one of the driving things that he changed in our platform was the ability to dynamically set up and change the computational backend. So we've at first started with an elastic MapReduce approach. And now because of Zechen, we have the ability to dynamically set up, or request GPUs, CPUs, and an unlimited amount of memory, that type of thing, because of his project. So, yeah. So I think I just wanted to quick highlight, I think with respect to opportunities in general, I know that UAB has a server, and so it's pretty common in your grant applications that you're making or your resource applications to budget in costs that are related to using that server, or reference that server when you're outlining your section of why you are capable of doing what you're proposing, right? And why you have the tools and resources at your disposal. One of the things that you can also include in there is cloud computing. So whether it's the BioData Catalyst platform or any other platform, right? Or whether it's the precision medicine platform, you can include that in your grant and your budget funding. And so we're happy to help with writing any of that. So that includes like the cost of software and compute resources. So you wouldn't necessarily have to buy a SaaS license for your team, let's say, or a MATLAB license. It would be included with your precision medicine account. And the way that we've kind of set that up is, especially for early careers, we try and make it as affordable as possible. So rather than looking at thousands of dollars a year for a license, or license fees. And we're hoping that all of the compute, we make all of the compute and the licensing all part of the platform. And for early careers, it's, I think, $500 a year. And for, I guess, not early careers, it's $1,000, I believe. And then some of our grants just come with this automatically. So Zedchen's grant, for example, are institute level grants that are really designed and focused around machine learning, image processing, genetics. Those automatically come with a precision medicine platform workspace and all the credits that you need to pay for it. And so if you're interested in opportunities in general at all, these are specific to the AHA. Obviously, with the NIH, you can still do this top section or any other grant that you're applying to. You can still put that in your technical budget. But for the AHA opportunities, if you're interested, if you go to heart.org and then there's a professionals tab, you can click research for application information. And that's really just kind of overall, all the application information that we have available for the AHA. Then there's also specific to the Institute for Precision Cardiovascular Medicine, which again is more data science driven grants, or I don't know if data science, but computer science, computational focused grants. And so those are under the Institute for Precision Cardiovascular Medicine. And then if you are interested in some of our data sets that are available, if you go to the Get With the Guidelines section, that has overviews of their different data sets and the proposals that you can make. And when your proposal has been accepted, you're automatically set up with a workspace.
Could I chime in a little bit, Laura? I think Jen mentioned the AHA right now hosts probably the large volume of COVID-19 data, right? Related.
Yeah, we have a, so if you go to this, I don't know if I can click, sorry, the very bottom third bullet on this page right here, to Get With the Guidelines, there's a COVID registry. And so we have, through, I guess there's a couple things, through Burst IQ and really through GitHub in general, we have access to, all of the public data that's been published relating to COVID-19. But then we also have our COVID registry data where we have our registry, which is the AHA's registry. I think we've got about 40,000 cases of COVID-19, records of patients with COVID-19 in hospitals across the country. And there's about 2,000 variables. I'll demo that when we get into it so you can see what's in it. And so you can make a proposal. You can submit a proposal for a research topic that you'd like to do. The committee reviews it. And then if it's approved, all that process is free. Then if it's approved, you get a workspace with the data and a mentor that helps you with the data as well. And you can propose things where the AHA data science team will help you with your analysis if needed.
Yeah, and we do
also, sorry, go ahead.
Yeah, I was going to say, and we do periodically run data challenges. So we had our first successful COVID-19 related data challenge where there was, you could participate and put up your research for a reward, at the end of it if you're selected. So we have opportunities like that come up periodically as well.
I don't know if Wes, if I have the link just off the top of my head, but if you have the heart failure link, would you put it in the chat? We're currently running our heart failure data challenge. It's open.
Let me grab it.
So when you register for the challenge, you'll get a workspace and you actually have access to the Get With the Guidelines heart failure registry. So I think it's, I don't know, a couple million records of patient records with heart failure from hospitals, again, around the country. It kind of ranges between, I don't know, anywhere from 200 hospitals to 4,000 hospitals, depending on the registry.
And I just dropped that in the chat for everybody to look at.
Cool. A quick question regarding the budget for early careers. So is this, for example, if I have a existing grant, can I budget for next year's and at this? Or you mean that was something brand new and we have to mention that AHA, PMP, and mention that and budget that in a new grant?
So it kind of depends on the grants. The institute grants, this one and this third bullet right here, those institute grants, that's one of the grants that you're funded with, Zechen. And those grants, since they're like specifically, grants that target questions around machine learning, genetics, use of data, basically, they come with, typically as part of the award, you're awarded a workspace and all the costs associated with it. If you, Zechen, came now as just an early career that you'd like to just use a workspace, either just for your team without a grant, or you were part of an AHA grant that wasn't part of the institute, then you can include that in your grants. So if you're a basic scientist, or some of our other basic science clinical grants don't necessarily get directly tied to a workspace, but if you would like one, you can budget that into your grant proposal for about $500 a year. And so either whether you're putting it into a grant proposal or you just want to use it in general, like use the cloud just in general and all of the software and the computing that comes with it. For early careers, it's $500 a year. Okay, so Wes, do you want to just go ahead and tell us a little bit about like the overall infrastructure and the system design of the platform?
Yeah, I'll give an overview of the and focus on the P&P workspaces. So, so this is a representation of essentially a the development environment, the analytical and development environment that we have in the P&P. And each of these workspaces it runs in its own AWS account. So at kind of the architectural level, everything is contained in a single AWS account, and it's actually designed to be more or less your own sandbox. That is, research sandbox that is cordoned off from the rest of the platform. You could blow up your workspace and it wouldn't affect anything else in the platform. So it's really for you and your team to run experiments, to leverage the power of those different instance types depending on what kind of research you're doing. So like Laura mentioned, we started early on with an EMR based workspace, which is an auto scaling cluster. You have to use Spark to run it, and it scales up and scales down instances based on the load. So that was really the one offering that we had to begin with, and then over time we got feedback from researchers, like you, Zechen, who expressed a need for other types of compute infrastructure. So over time we evolved our offerings to match up what the research need was. So we started offering one just kind of simple EC2 instance. If you're not doing a lot of compute intensive work, you're doing data exploration, maybe some basic statistical analysis, that kind of thing, versus a more higher powered GPU instance where you could be running intense computations. You could be doing things like convolutional neural nets or other highly intense repeated computations and need that kind of processor. But though the workspace has different footprints in terms of the computational infrastructure, the experience of using the workspace is essentially the same across all three, in terms of you've got a Jupyter notebook, which you all would be familiar with. You have the option of using RStudio. We'll soon have MATLAB available, and then optionally you can also use SAS. So especially with a lot of the internal researchers that use the P&P within AHA, we have SAS, and we can make that available to any team that comes on and wants to use that particular package, set of tools. So in addition to kind of the basic more or less IDE offerings, we have standard packages that have been installed in the workspaces by default, like this isn't an exhaustive list, but it just gives you an idea, like TensorFlow with Keras, Shiny for building R-based applications, Plotly, Pandas, for doing your plotting and data manipulation. And we have a whole set of other tools and packages that we publish on the website, and you can take a look at it. But essentially, we over time we grow the standard offering. So when you spin up a workspace, you don't have to go and install TensorFlow yourself or Shiny. It's there. You have the ability to install additional tools and packages, but there's there's kind of like a gold standard base that everybody starts with. And like I said, our philosophy is to make this environment not super opinionated in terms of the area of research, but to make it an open environment where we can evolve, based on the needs of the researchers that we hear, in our feedback, in our forums, and enhance the offerings over time that are, as you all know, constantly changing. So we can take advantage of both the open source community, but also the evolving offerings from AWS, in terms of services and instance types and things like that. So that that kind of is an overview of the workspace itself, and we get access to it through through an AppStream session. So it's more or less like a browser in a browser. That's the way that we're able to securely provide access to our teams, and you can either connect to the data lake, the data that AHA has hosted. Also, there's some limited public internet access. We have full control over that. But also you can, of course, like usually is the case, you bring your own data, either upload through the portal itself, through your browser, or you can optionally sign up for an SFTP repository, where if you want, more of a static connection, where you might be loading more data, if you're going above like 20 or 30 gigs, if you're loading up to terabytes of data at one time, you can use SFTP, and that's a little bit, of course, use any standard SFTP client, like FileZilla or CyberDuck, or whatever you choose. So we have a few options there for getting data into the workspace itself, and all of these instances have EFS, extensible elastic file storage that back them all up. So you can switch back and forth between different instance types, but then stay connected to the same file storage that that will never run out, because it scales up based on what you actually load in there. So we have the data storage decoupled from the instances themselves. And that's really an overview. Any questions? All right. Great. Thank you guys. Laura, I'll turn it back over to you.
Thanks, Wes. So I just wanted to we have a few slides that kind of address some specifics about the platform, in the context of some of the points that I made earlier. So with respect to secure data access and accessing licensed software, you'll see a lot of this when we do the demo. But there's ability to request data, that is either from another institution. So we do have genetic data sets and other clinical trial data sets that are available just by request through the platform, and the AHA just acts as a third party to manage the distribution of those data sets. And then we also have our AHA data assets, which are the get with the guidelines data sets, or research goes red data. So for the heart failure registry or the COVID registry, that you can request access through on the platform, and I'll show you that. But then you also have the ability to transfer your own data in. So if you have data from your institution, you can SFTP that in, which does create an SSH connection. And you can also just upload probably up to 20 gigabytes pretty easily just through upload. It gets really dependent on your internet connection, but if you're uploading a lot of data, sometimes the SFTP is a little bit easier. And then when you do that, basically the data is only accessible to the people who have access or who have permissions. So if you have a workspace set up and it's your workspace and there are no protected data sets in there, The only people that can access the data are you, unless you choose to share it. And then if you were to share your workspace with, let's say, a colleague, you can add them to your like DAC or your IRB for the AHA data sets that are owned, so that those get approved before they can gain access. But then really just the people who have access to the workspace are the people that can access the data. And then within that workspace, there's a variety of different tools, including licensed software. So right now, for example, all of our users get SAS, so they can use SAS. And then we also just are currently launching MATLAB. So they kind of built like a custom setup for us. So we have the option to use MATLAB as like a G kernel, and also have access to the MATLAB like interface, the IDE within the platform. And then on top of that, we have access to open source softwares like Jupyter Lab, and R. So you can come and share those, I guess, work in those environments where the environment is all pretty standardized. So everyone on the project is accessing the same environment. And we can actually set up, or you can too, you have the options to set up like a Conda-based environment if you want to have a specific environment that's similar that you already have set up. Or you want to manage your environment using Conda, you can do that for different projects. And then also, SnakeMake for larger projects or projects that require more compute resources, all of that is available as well. And then so the ability to collaborate is, as I mentioned, it's basically just sharing your workspace. And then you can grant approvals to data or get approvals for people if you have not already gotten them when you requested it. If there is AHA managed data available in that workspace. And so you can just click the share button here, and then you can add collaborators. And so it's really nice for projects that are either ongoing or have multiple students, is you can add a student when they come on and then take them off when they leave. And you can just continue to do that so that you don't have to worry about where the data is, where the files are. They're all just accessible in the workspace. And once their access has been removed, they no longer have access to the project. And then you also have within the environment, you're sharing files, and you have the ability to kind of control the version. So some of our PIs find that really nice because they can control which version of the data or subset of the data is accessible for the project for the users that are on the workspace. And then, as I'd mentioned, the access to compute resources. So currently what we do is we kind of just offer these like 3 standard setups. And then we can scale up from there. So we currently have the option of just a CPU and regular memory setup. So this is pretty much just like a standard computer that you would get. And then additionally, we have that set up, but with a GPU. So you have the availability to use a GPU if needed. Both of those can scale up in the number of CPUs. I don't exactly know if there's a limit, but I think you can go up pretty much as high as you need to, like in the 264 range as far as CPUs go. And same thing with memory. And you can also have multiple GPUs. So we have a couple of teams that are working on workspaces that have 4 or 6 or 8 GPUs set up right now at once. And then you also have the access to an auto scaling cluster, which is just a cluster of workspace instances that scale automatically using Spark. So if you're using certain machine learning, it works really well with Spark, and it will automatically scale your process so that it will run as optimally and as efficiently as possible. So if you're using Spark-based packages, then definitely the auto scaling cluster is the way to go. And that's also scalable. So you can scale up to an auto scaling cluster that goes above 32 CPUs, for example. And then just with respect to reusability, what we've tried to do with the platform is with all of our data that we provide access to, you get access to these data usage tutorials, so you have an idea of how to use the data in a variety of different languages. We almost always provide them in R, SAS, and Python, and we would help if you needed other languages as well. So these just kind of show you how to just kind of quick, get started with the data that you've been approved for. But they are also just plug and play. So even if you don't know how to code, you can run them. All the code is there for you as well. And we have that kind of extended to tutorials themselves. So we have tutorials in the platform that are as simple as just learning how to set up a software or install a software, or use a basic package, but then can go to more complex levels of analyses, like learning how to perform a GWAS analysis or the quality control for genetic data, processing for image data, that kind of thing. And then we also have the ability where users publish their notebooks when they're done or their work when they're done. And then you can copy those directly into your workspace and run them as well.
Hey, Laura.
Yeah.
Ralph had a question about software costs, software licensing costs. So
Yeah.
he was asking if there was an additional charge for MATLAB, SAS, etc. So it depends on the particular piece of software, you know, what the licensing arrangements are. But for SAS, that comes with usage of the workspace, right, Laura? There's no additional fee.
In general, we try and set that up, you know, set up the workspace that way.
Right.
The MATLAB, the way that it's set up right now is that as long as your institution has a license, you can use that license even if it's not necessarily transferable to the platform. MATLAB will allow it, so you can use that license in the workspace. We do have a few licenses that like we can use temporarily, and then MATLAB has the offering where you can get the license for the whatever project you're doing. Get the stuff that you need. So most of the researchers that we've had, their institution provides that for them already, and then they just bring, you know, they use that key or that login information in the platform. But we would work with you on a case-by-case basis if that was not the case. And then I think you also had a question about, does that go to direct or indirect costs in the grants? I think the platform cost it kind of depends on the grant that you're setting up. If you have a pretty, you know, heavily computational or data oriented grant, I'd probably put it in direct costs. And then with respect to, I guess, licensing, as I mentioned, the majority of software already is covered, so you wouldn't need to add any additional. Right now, there's the only case of MATLAB, but I suppose that could be extended in the future to other softwares.
Thanks. I'm just trying to get, you know, understand it, because, you know, it's something that comes up and I don't have the answer for it. And even, you know, I was going to ask a question earlier when we were talking about lower costs in the cloud. I keep looking for a good study on that, because in things in various national groups like at CASC and PERC and SC, I'm always seeing studies where, you know, there's a certain economy of scale, and if you have the infrastructure set up, it's a lot cheaper on-prem than in the cloud, given the nature of how often some of the, it depends on the workload, right?
Right.
And it's hard to answer because there isn't clear data. And even when you go to Amazon or Google Cloud to try and price it out with like-size systems and try to factor in, you know, depreciation of capital and all of that, it's rarely an apples to apples comparison.
Yeah, their calculators don't factor in the true total cost of ownership of, you know, moving workloads to the cloud. And that's, you know, that's probably one, you know, talking about challenges that Laura was talking about before is, you know, it's really a matter of when you're investing in the cloud, getting out, you know, what you expect and being able to predict what your investment will be. You know, of course, the model is great of being able to, you know, pay for what you use, but, you know, am I paying the right amount? Am I using the right amount? Am I using the right services? You know, being able to weigh all of those factors, and that's what we're you know trying to do here in the platform, in the PMP, is to simplify, you know, that calculation, and to give more predictability around cloud spend.
Yeah, I think that was like a heavy motivation. I mean, in general, that's the objective of the platform. But it was also a very heavy motivation into why we kind of tried to just set these, like, it's $500 a year, and, you know, the AHA will either cover the extra costs or figure it out for the early careers, because we have a strategic partnership with Amazon. So that helps us with our costs in general. And then, so I think that's the motivation behind setting something up like this. But the other thing that we have here, and I think we'd probably have a hard time speaking to other platforms. We might be able to speak to AWS, but that's also why if you just want to, if you go to precision.heart.org, where the platform is, you can go to just about and then contact us. And if you are, writing a grant, that is one of the services that we offer just at the AHA, where we'll help you write out a budget for, accounting for that based on your requirements. Because we do that for our grants that the AHA applies for, and our funders, big funders, we do that for them, and include our use of the platform in that. So we have kind of some more standardized approaches, but it is also the reason why we tried to just set up this $500 a year.
And Ralph, to add to this, we worked with Google Cloud. This service is terrible, and that's why I invited Laura and Alison here. So I think both PMP and the 7 Bridges or the BD Catalyst, they are targeting the researchers, and they are really research friendly compared to the commercial cloud like Google Cloud I worked directly with. That's terrible, and we spent probably 2 weeks or 3 weeks to allocate a node. That's terrible. And this PMP and the 7 Bridges, the platform is quite versatile and it's functional. So yeah, Laura, Ralph is our assistant vice president for the research computing team, and he's the boss of the IT here. So
and we're trying to strike the balance of on-prem, and we have significant on-prem resources. I mean, we're kind of a private cloud.
Tell me a little bit about your server.
And it's that balance. Like a question that keeps coming up too is that we don't recharge for services. So from a development perspective, a lot of faculty like using the resources here because they can be creative without worrying about a process run amok.
Yeah.
And then from my end, want to understand then what it would take to transition whatever they develop for. And it's interesting because I see a real value of some of the of these type of approaches to that data. I'll just call it data harmonization, in the sense that you start to have some standards for the data, and you can actually maybe have some apples to apples analysis going on. But then there's also my favorite Einstein quote: "If we knew what we were doing, it wouldn't be called research, would it?" So people inventing the latest and greatest stuff want to be able to have full flexibility and control. And again, it's that that's never going to go away. That balance of production versus R&D.
Yeah, I think one of the things that we, we definitely struggle with that balance all the time as far as, and I think probably BioData Catalyst does as well. I think, one of the things that might be worth investigating if you wanted to, you can either contact us, this will go to me, I will figure out, who the people to bring in. But we do offer kind of like we have a couple of institutions that have white labeled the platform for that type of service. So their institution has their own cloud that they can come to instead of using their high performance, their hardware that they're currently managing. It's a little bit of a cost. It's a completely different cost, setup and structure. This presentation was a little bit more geared towards, individual grantees at an institution or PIs trying to apply to a grant. But if that is something you are interested in, and there's ways that Wes and I can help, we would definitely be happy to set up, a meeting to just even just lessons learned, or help you, if that's something you are interested in transitioning to at university, so that you have more information as far as what the actual cost is. We'd be happy to set that up as well.
Yeah, it's almost like when your organization does set out to go to the cloud, you almost need to set up, your own data lake just to capture the cost. What services are you using? how much are they, who's using it, over time, what does that profile look like? So you can actually make, intelligent decisions about either, moving additional workloads to the cloud or moving things off, moving them to on-premise because maybe it doesn't make sense, like you were talking about, Ralph. So it's not, it's not yet a science. It's very much an art, making those kinds of decisions. And it sounds like you've seen that, firsthand, Ralph. So that's where I think it's going, is being able to make, the cloud providers are going to have to make the case more and more for moving to cloud when they get, when we start seeing data come back about what the actual cost is, how much these organizations are investing versus what they're getting back. And that's going to make, they're going to have to adjust to that, more transparency, and that will factor into decisions about, cloud adoption going forward. It's going to be interesting.
Yeah, and it seems like right now there are two. We have a controlled experiment going on, because in the more of the life sciences NIH space, it seems to be in the cloud, and in the physical sciences NSF world, it's all on-prem.
on metal.
So we have an interesting experiment going on for the next few years.
Absolutely.
Ralph, I think a practical question is, so we really need cloud, because, for example, we perform some huge computational work in genomics, de novo assembly, these tasks. Our Chiha, the big memory machine, actually cannot satisfy our needs. So that's why we contact the Google Cloud and see if we can request a large memory node to perform the de novo assembly tasks. So
so now that the need has been, and that's again that balance, right? So there might be projects that are large enough that will need to burst to the cloud, but to meet it's only really been, so I think of it as maturity, and in fact that's kind of the buzzword right now, measuring your institutional maturity around research computing services. And we haven't had a lot of demand for that sort of resource. That's been increasing in the last year, and we're adding major resources that enable that. Will that solve everything? It's that balancing act, trying to stay ahead of the demand. That's one of the values that one of the value adds that you get in the cloud. It's very easy to add resources, but there's a price, you need to be able to anticipate what your need is going to be and have the budget for it, as opposed to just being able to use it, when it's on campus, and not worry about what it's going to cost you.
Yeah, I think, in general, that's kind of the way we set up this. I think for Zechen's case, I guess, I don't know if his grant was around when we finally got this. He was probably still almost close to done. We finally got all this working, but that is also one of the reasons why we have this kind of basic, I guess, default setup, is it's just, basic resources that don't really necessarily require a lot of extra funding. And we find that this kind of works for 80% of our cases, right? So then we scale up, add a lot more memory or add a lot more CPUs as needed. And so I suppose it would be, it'd be interesting to see, if the cost of this would be equivalent or maybe more, but then for those 20 cases, you could come to the cloud or not, there's lots of different options.
Well, I mean, I think our approach is we're setting it up so that, again, the R&D can happen on-prem, and using the same technology. It's the same spin up on the fly, allocate resources automatically. It's coming live next month, that type of a resource. But again, and what that does then is that if it's a project that is going to be national, if you will, it can get moved up to cloud resources. Just move the container up. Might require some tweaking, but on the whole, we've had several successful projects that were developed in containers on-prem and then moved to a cloud resource where
We do have quite a
needed.
few grantees that use that system now, where they bring their containers and Conda environments to set that up in the workspace.
Can
and
do
then they-
that too. Yep, that's possible as well.
Where they, but they
I mean,
do,
that's the beauty of it, right? If you're developing your applications that way and taking a much more software engineering approach to developing the applications, it's, I say it with a with a grain of salt, because nothing's ever perfect, but we've not had to spend months moving an app. So far, of the half a dozen or so that have gone that route, it might have been a few days of tweaking, but that's about it. And some of them have been an hour. So it really depends on how well the application was developed.
Yep. The goal is to do it better the next time, based on what you learn. And that's,
well, yeah, exactly. And I think, a credit to all the people who've been attending this conference today. They actually are spending a fair amount of effort with, I'll say, professional software development approaches and algorithm approaches, and that's helpful.
cool.
Yeah. So, well, I guess I should. Maybe I'll just quick demo. We talked about a lot of the basic functionality of the portal page. So what we call this the portal pages, and it's the portal to the workspace or your computational environment. And so when you create an account on precision.heart.org right here, just click this, you'll be able to enter your email in. And then you have the option to request a workspace or request data, or you will be automatically provisioned if you're set up as a grantee, but you'll have workspaces. And when you click this workspace tab here, this is kind of the page that you'll see, and this is where you can actually share, as I mentioned here, where you can add users, take them on and off. And it's just entering in an email. They'll get an email either telling them, if you enter an email and they have not registered for the platform yet, they'll get an information, just a notice saying, the workspace is ready for you, and then they can come and create an account. Or they'll also have the ability to, if they already have the account, then when they log in, they'll be able to see it. And then here you can, upload your data. You can also request SFTP. So if you have an SFTP set up, it'll just be displayed here on your portal page, and you can do that under the requests. And it will actually walk you through that when you try to upload data as well. So for example, if you're just clicking upload here and you need to, if you have large files, this will take you to the SFTP request page. And so then when you actually click this launch workspace, what that does is it brings up a computational, it's weird that it's displaying like that. Okay, here we go. What that does is it brings up this virtual computational environment that you guys have shared access to. So within that environment, there is Jupyter Notebooks itself. So that's probably very similar to what you guys saw with BioData Catalyst. I believe they have most of their stuff is Jupyter. And then they also have, we also have Jupyter Lab here. So it's just a little bit more development friendly, I suppose, as far as, access to, you can use a Python script in here, for example, like a .py file or a .r or .sas file in here, as well as any of the Jupyters with the kernels. So you can, and then you can also, start those consoles. And you have access to the terminal, so you can use the terminal here for command line tools. If you just like to program in the terminal, you also have access to the terminal through Jupyter. It's also where you can install. So you have root access in here, so you can install anything you'd like. Conda is set up, but you also can bring your own. You can install all of your own versions of things as well, and then you can install anything that you would like to, as far as installing Python or RStudio. You have pip install and install packages, so you can install any of the software packages that you'd like. We also provide technical support for that. We have, for the most part, most of our users have been satisfied with this, but if there are specific IDEs or other things that you would like to use, we can help you install those and get those set up. I think our main cases have been with I guess mostly like database type. We do occasionally get requests for like VS Code or PyCharm. We do have a Windows version of a workspace that kind of allows having those types of IDEs available within the workspace as well. And then SAS Studio and RStudio work the exact same as your personal computer does. It's going to look just like it does your personal computer. And then, as I mentioned, MATLAB is coming. So pretty soon you'll see in Jupyter Lab, you'll see the MATLAB icon here. So you can open a Jupyter Notebook, and then you'll also be able to see a MATLAB tab up here that works where you'll see the MATLAB IDE. So I think, in general, that kind of covers the basics. One of the things that I'd like to maybe highlight a little bit, if you have questions about the software that is available, we're currently releasing a new update to this page, but if you go to resources and then about workspaces, you'll be able to see all the software packages that are available MATLAB and SAS are not listed. We're sent, those are the updates that are coming. But other than that, they see all the standard packages that are already installed. If you have questions about those, you can also request, when you request the workspace, for certain stuff to be installed when your workspace is provisioned. And then also under the resources, we kind of have this page here, this usage and docs page, where this will bring you to kind of this home page. If you questions about how to request a workspace, like which architecture should I select, or what are the steps I need to do to get a workspace? This is kind of just like a good step-by-step guide. The user guide here, this will bring you to, in general, general questions like how do I use GitHub in the workspace, or how do I, upload files, get files out, access data or repository, anything like that. What's my software password, stuff like those. All that stuff is provided in this usage user guide. And then additionally, we have these data resources that we've recently been building out. So if you were to request access and get approved for a proposal, let's say for the COVID, to get with the guidelines COVID registry, you'll get access to this page here. And what comes with that is the usage and guidelines also are in your workspace, but you'll be able to see it, the tutorials for how to use the data in the variety of different languages that you would like, where the data is stored in your workspace. All of that. Also, each page has like a getting started with the data. So it kind of tells you just the very basics you need to know about the registry, where it is in your workspace, what folder it's in, links you to the user guide, all that kind of stuff, your basic passwords. And then we also provide this table here where users can actually see what's in the data. So for the COVID-19 registry, for example, you can see all the variables listed here. There's an overview summary that's plotted here, like with your, not really the best example, but like here for discharge status, when you hover over here, you can see like, the quantity or the percentage that are discharged to another health facility. And when you click this arrow here, you'll actually be able to see the breakdown. It's basically like kind of like a table 1 of all the variables that are available. And then this will directly link to any information related to this variable. So where it is in the collection report form, the design or collection, the clinical trial protocols that are around it, if there's anything like that related to the variable, if there's logic associated about how it was derived. So if it's like a derived variable, that will also be, so race is a good one. Sorry, it's a little weird view. So if you were to go to race, there's all these different race variables that are available. So if you have questions about how this, compiled race variable was put together, you could look here in, these are the categories that are available in that variable. And then if you want to see the logic of how they're derived, it's here. And we also provide just analysis guidelines. So if you were to look here, we have definitions and logic for derived variables like defining mortality, I don't know, COVID composite outcome, that kind of stuff. Information on just the pre-processing or what cleaning has been done, that type of stuff. The exclusion criteria, how to handle missingness, things like that. So those are all provided for the in your workspace, you can use them, and then you also have access to this site here. So if you're a PI on the project or you're working with a clinical, expert, then they can come here and they can just click around and see things. You don't necessarily have to know how to code or use the data. And so, that's kind of the basics. I think I'll, pause there, see if there's questions. Feel free to, let's see. I don't know if I saw if there's any other questions in the chat, but if there's any questions regarding anything, happy to answer them. Again, there's just under the, let's see, the about here and contact us. You can always contact us and we'll answer any of the questions that you have or help you, I guess, with your budget on your grant or institution level, as we said with Ralph, anything like that.
Great, thank you, Laura. Thank you, Laura and Wes. And also, I would like to thank Alison and Dave. Thank you so much. And I think we have the recording for this video, and people who haven't seen this but registered this meeting can still review this later. So I think these resources are very helpful.